My name is Neil Winegarden. I'm a field application scientist with 10x Genomics, and I'm going to be discussing some practical considerations and best practices for single-cell and spatial transcriptomics. Traditional gene expression assays have relied mainly on bulk analysis, which give you a general idea of the expression happening in a tissue, but lack cellular or spatial resolution. Single-cell gene expression assays allow researchers to characterize individual cell types within a tissue, providing much greater resolution and understanding of the underlying complexity of the tissue. Now, with spatial gene expression methods, researchers can map the spatial pattern of gene expression. Integrating these two data sets provides key insights into how tissue functions. By combining single-cell data with spatial data, researchers can now address a wide range of biological questions, including refining cell atlases, discovering new cell types with anatomical location, characterizing tissue and cell architecture across development and disease, and identifying pathological processes. I'd like to start with a very brief introduction to both single-cell and spatial transcriptomics approaches. In order to study transcriptomics at the single-cell level, it's critical to partition the cells into discrete reactions in order to isolate and tag the transcripts from each individual cell. The more partitions that can be created, the greater the level of heterogeneity that can be assessed. In our approach, individual cells are isolated into discrete aqueous drops within an oil emulsion. These droplets also include a bar-coded gel bead that provides tags that are incorporated into the resultant cDNA so that transcripts can be mapped back onto the cell of origin. Typically, up to 10,000 individual cells are captured and profiled. We call these droplets gel beads in emulsions, or GEMs. The gel beads act as an oligo delivery tool. The beads provide the necessary reverse transcription primers that allow for creation of cDNA from the mRNA transcripts released when the cell lyses within the droplet. These oligos contain a poly-DT sequence that will hybridize to the poly-A tail of the mRNA, as well as a barcode which identifies the cell of origin. A unique molecular identifier, or UMI, allows for the initial transcript of origin to be identified, providing an accurate assessment of the number of initial copies within the cell. A microscopic picture of one of these GEMs is seen here. Normally, such a picture cannot be taken because the cell lyses too fast. However, we've changed a few things in order to get this image. As you can see, there is a discrete droplet isolated from the rest of the solution in which we have a gel bead and an individual single cell. This is the key to obtaining single-cell transcriptomes. The generation of tens of thousands of such droplets allows for many cells to be profiled individually. The resultant sequencing-ready library from a single-cell transcriptomics experiment contains the barcode sequence, UMI, and a cDNA insert sequence that allow for mapping of individual transcripts onto a reference transcriptome and the association of that transcript with the cell of origin. In a spatial transcriptomics approach, mRNAs are captured while maintaining their relative initial position within the tissue. The resultant transcriptomic information can be overlaid back onto the image of the originating tissue section to provide a high degree of spatial resolution of the initial transcripts. In our approach, a similar oligo construct is used, again with a poly-A tail to capture the mRNA. This time, the barcode provides the XY coordinates of the spot on the slide. Again, a UMI sequence allows for accurate assessment of the initial number of copies within each spot. Spots are arrayed in a hexagonal pattern, with each spot having a diameter of 55 microns and a center-to-center pitch of 100 microns. The total capture area is 6.5 millimeters by 6.5 millimeters squared. Multiple capture areas on a single slide allow for multiple tissue sections to be profiled. Tissue sections, typically on the order of 10 to 20 microns thick, are placed onto a capture region containing the arrayed oligos that I just discussed. The tissue is stained, imaged, and then permeabilized to release the RNA for capture. The RNA is reverse transcribed on the slide, and the resultant cDNA is released in order to create a sequencing library. The resultant sequencing-ready library from spatial transcriptomics experiments look essentially the same as from a single-cell transcriptomic library. It contains the barcode sequence, UMI, and cDNA insert that allows for mapping onto the reference transcriptome. This time, the transcripts are associated with the spot of origin. Now that we've discussed the underlying technologies, let's quickly review a few practical experimental design considerations. Many researchers first embarking on a single-cell experiment often ask about the appropriate cell number and sequencing depth required for an experiment. The truth is, this is a balance between the question being asked, the complexity of the sample, and the sequencing budget. A more complex tissue for which a researcher wishes to detect a small subset of cells will require more cells to be profiled. More cells will in turn cost more to sequence. As such, researchers need to balance all of these aspects. There are no quick answers here, but tools such as the How Many Cells calculator from the Satija lab, the staff at your core service provider, or the technical staff at the technology partner will be able to help you with this. The depth at which you sequence your single-cell experiment will in turn be related to the overall complexity of the transcriptome of the cells. Larger cells, like cancer cells with more initial RNA content, may require deeper sequencing depths than smaller cells with less RNA, for example, an immune cell. As with single-cell gene expression libraries, tissue complexity also impacts the required sequencing depth for a spatial transcriptomics approach. If the tissue placed onto the slide is less complex, as seen here with the human brain section on the right, it will require less sequencing to reach a similar saturation level than a more complex sample, such as the mouse brain section on the left. Of course, this is not to say that human brains are less complex than mouse brains. However, in any given 6.5 millimeter by 6.5 millimeter square, this happens to be the case. Another common question relates to the number of replicates that need to be run. Fortunately, these approaches are very robust. They are very reproducible at the technical level. Here we see a representation of the same sample run 16 different times for a single-cell transcriptomics experiment. These were run over 2 different microfluidics chips. It's fairly apparent that the overall signatures being seen here are highly reproducible. That same data presented in a more quantitative fashion shows that the technical replicates from the same source material are highly reproducible, each of these pairwise comparisons having R-squared values above 0.99. Looking at individual genes of interest also shows a high degree of similarity in technical replicates, in this case, even across different chips, which was therefore separate runs. Here, we're looking at biological rather than technical replicates. 2 sections of an E18 mouse brain were profiled separately. In such a case, we would expect a biological variation to be relatively minimal. Indeed, you can see a high degree of reproducibility with R-squared values above 0.95. Taken in total, I tend to recommend that your experimental budget be used to test true biological replicates, separate mice or separate patient samples, rather than focusing on more technical replicates, as these technologies really are quite reproducible. Spatial transcriptomics approaches are also highly reproducible. It's not really possible to do a true technical replicate with this technology because the tissue section really is consumed during the process. So here what we have instead are 2 serial sections of a mouse brain. For these 2 sections, the software identified 12 distinct clusters of spots. They were identified on the basis of transcriptional similarity. The spots are colored on the basis of which cluster each spot was associated with. And you can see the remarkable similarity between these 2 sections with very, very similar patterns existing. If we were to look at the expression of a specific gene, in this case myelin basic protein or MBP, it is again evident how similar the patterns are across these 2 sections. So taken as a total, each of these technologies was really quite reproducible at the technical level. Now that I've talked about some of the experimental design considerations, I'd like to talk about considerations for sample preparation. In order to ensure the success of a single cell or spatial transcriptomics experiment, the quality of the sample must be optimized. A good quality sample is critical to overall performance. When working with single cells, it's critical that the tissue be fully dissociated into a single cell suspension. This not only ensures that the microfluidics in the system will not become clogged, but it also ensures that each partition will have a chance to capture a single cell rather than a clump or aggregate of more than one cell. Cell viability is also paramount. Dead and dying cells will also be captured, and this potentially leads to spending some of your sequencing budget towards cells that are not really of interest. Perhaps even more problematic, dead and dying cells are fragile and they can lyse very easily, releasing RNA into the ambient environment. And this creates a background noise that is hard to distinguish from the true signal of a cell. It is therefore critical to treat cells gently, wash them gently, and produce a high viability single cell sample. The removal of debris and other cellular aggregates will help to prevent the clogging of the microfluidic channels, as well as reduce the noise in the transcriptomic data. We recommend filtering or straining these cells. Accurate cell counting will be critical to providing a consistent cell target number. Automated counters can make this process a lot more easy and reproducible. Finally, the cell storage and the efficient processing of those cells are key to ensuring that the viability as well as transcriptional signature of the cells are not impacted. Our website, support.10xgenomics.com, has a cell preparation guide which you can download and discusses each of these topics in even more detail. Similarly, when working with spatial transcriptomics approaches, it is important that the tissue be preserved, in this case frozen, in such a way that both histology and RNA quality are preserved. Again, we have a demonstrated protocol available on our website at support.10xgenomics.com. That guide discusses all of the key concepts of tissue preparation. Some of the key topics are RNA quality assessment, tissue freezing, embedding, as well as sectioning and placement of the tissues onto the slides. The specific method of freezing is critical, as it ensures maintenance of tissue morphology while protecting RNA integrity. In general, we recommend a rapid but very even freezing method. Specifically, isopentane in a liquid nitrogen bath, which prevents tissue cracking during the freezing process. Frozen tissues are embedded into OCT for proper sectioning and placement onto the slide. As I said, rapid freezing is critical to maintain that morphology and RNA quality. However, directly freezing in liquid nitrogen can be problematic, as it can cause tissue cracking. The low boiling point of liquid nitrogen causes rapid expansion of air pockets within the tissue that can cause cracking of the tissue and disruption of the overall morphology. With spatial transcriptomics approaches, we highly recommend performing an RNA integrity assessment, obtaining the RIN value or RNA integrity number. This helps to ensure that the sample has been optimally stored to ensure good quality libraries can be produced. Again, our tissue preparation guide provides details on how to do this. In general, we found that RIN values of 7 or higher are associated with a high degree of success. At the beginning of this talk, I mentioned that there is a strong synergy in combining single-cell and spatial transcriptomics approaches. So I'll quickly touch on a couple sample prep recommendations for such an integrated analysis. With our current spatial transcriptomics approach, which I've already talked about, we work from frozen tissues. For researchers who are interested in an integrated approach looking at both single-cell and spatial transcriptomics, it is possible to perform single-cell transcriptomics from a frozen sample. Doing so requires isolation of nuclei, as it is not possible to isolate intact viable cells from a flash frozen sample. With a nuclei-seq approach, There are fewer overall transcripts as the cytoplasm is being discarded, but this does allow for the direct comparison of single nucleus data with spatial data from within the same tissue. Although single nuclei approaches are somewhat less sensitive for the reason I already discussed, the ability to identify cellular subtypes is still largely preserved. You can see in this example here, two samples that were of intact cells that were isolated from neuronal tissue and two nuclei preps. And you can see that the overall patterns are very similar. Looking at individual cell markers from both intact cells and from isolated nuclei shows that in most cases, canonical markers of individual cell types can still be detected and the cell subtypes identified. Once more, our website has a number of demonstrated protocols for the isolation of nuclei for single nuclei sequencing, depending on the starting tissue type. We have guides working from tissue culture or suspension cells, easy to dissociate tissues, or more complex or frozen tissues. The overall process is relatively straightforward, and there's no requirement for dissociation of cells before nuclei isolation. Nuclei can be isolated directly either from fresh or frozen tissue. Dr. Luciano Martelato, now the scientific director of the Single Cell Core Laboratory at Harvard, has made public a protocol that he and his group developed that they called the Frankenstein protocol. This takes nuclei isolation procedures one step further, using flow or FACS in order to purify isolated nuclei. This protocol can be found on our website in the Customer Developed Protocols section. Dr. Martelato has also developed an extension to this protocol called the Daughter Frankenstein protocol, which uses gradient separation rather than FACS. That protocol is available on protocols.io. When preparing nuclei, a microscopic assessment of the nuclear membrane integrity can be of particular value. This slide refers to single-cell attack sequencing, but the same premise holds true for gene expression. Nuclei with more intact membranes will typically provide higher quality data. I'd like to close out this presentation by briefly talking about informatic approaches to integrating single cell or single nuclei and spatial transcriptomic data. Recently, there have been a number of publications that bring together single cell and spatial transcriptomic information. This sort of integrated analysis provides even greater understanding of the complex biology underlying processes within tissues. The overall concept generally involves using the transcriptomic signatures identified for various cell types in a single cell experiment and finding those same signatures within the tissue in order to identify where these cell types exist within the context of the intact tissue. Here you can see a couple methods mentioned: the robust cell type decomposition tool from Rafael Irizarry's lab at the Dana-Farber, and Seurat from Raul Satige's lab at the New York Genome Center. These tools can actually work with datasets that were generated from different tissues, although from the same type of tissue. However, they can be even more powerful when analyzed using data from the same tissue. A practical example of the advantage of integrated analysis can be seen in this paper from Maynard et al. At the Johns Hopkins. Using 10x Genomics' Visium spatial transcriptomics platform, the researchers looked at the dorsolateral prefrontal cortex of the brain, and they were able to identify the key cortical layers based on gene expression patterns. Overall, it was possible to identify 6 cortical layers. Key marker genes were used to help define the spatial topography of the overall expression in the 6-layered dorsolateral prefrontal cortex. From there, adding single nuclei RNA expressions datasets generated on the 10x Genomics single cell expression platform helped to provide further insights as to the function of molecularly defined cell types. It was possible to use both of these datasets to register the single cell expression patterns into spatial contexts and to identify which cortical layers each cell type resided within. Here's another example, which involves some of our own 10X scientists, including Dr. Wyatt McDonald. In this case, the same triple-negative breast cancer sample was processed with a Visium spatial transcriptomics assay, as well as our single nuclei gene expression assay. Integration of single-cell and spatial data was performed using Seurat from the Satija lab. You can learn more about how Seurat does this at the link provided in the last bullet point here. Single nuclei sequencing produced a robust atlas of cell types, from immune cells to stromal cells and tumor cells. The signatures of each of these cell types were then used to integrate with the spatial transcriptomic data. Here we see the localization of tumor-infiltrating lymphocytes, specifically T cells, in these tumor sections. Pathologist annotations of the tumor sections are seen in the panels below. I'll point out that the largest region, encircled in the darker green shade, is annotated as invasive carcinoma, and the blue regions were identified as immune cells. Here, you can see that indeed there are T cells within the immune compartment. However, it also appears that there seems to be some infiltration of these cells into the tumor. When looking at B cells, we see that these cells are more isolated to the immune compartment. Very few of them seem to be invading into the tumor, as we saw with the T cells. The researchers also looked at a number of other cell signatures. In this case, we're seeing the signature for a luminal progenitor cell type, which can be seen throughout the tissue. Now, lest you think this only applies to human or mouse systems, this is a wonderful example from Montre et al. Studying the developing chicken heart. Tissue sections from various points of development were taken and profiled using the Visium system. Similarly, tissues were dissociated into single-cell suspension and analyzed using the 10X Genomics single-cell platform. This data was then integrated and analyzed. Throughout the stages of development, a number of cell types were identified to create a diverse cell atlas across a developmental time course. The proportion and distribution of the cell types changes over this developmental time course, starting with more progenitor-based lineages in the early stages and progressing to more highly differentiated and mature cell types such as cardiomyocytes in the later stages. We can also see that there are regional localization of these different cell types within the tissue architecture. As I mentioned before, the spots on our spatial transcriptomics platform are currently 55 microns in diameter. As such, one spot could contain more than one cell or cell type, theoretically between 1 and 10 cells. Typically, it's more like 2 to 4 cells. New approaches are emerging that allow for a deconvolution of the signature within a given spot. In this way, it's possible to estimate the total contribution of individual cell types. This slide highlights a few of these methods. I'm going to show you some data from a couple of these approaches. M. Freg from Alexandrina Goeva's lab at the Broad, and Spotlight from Holger Hein's lab at the CRG in Barcelona. NM-Freg is being used here to deconvolute the signatures within the hindbrain of an adult mouse. Specifically, in the left panel, the spatial distribution of parvalbumin interneurons is being identified, and on the right, it is signatures for Purkinje cells. Both of these panels are colored by a temperature scale, which shows the fraction of each spot that contains signatures for that cell type. In this example, Nieto et al. From the Hein lab were looking at the tumor immune microenvironment. In the left panel, you can see that a number of cell types were identified using single-cell transcriptomic analysis. For this analysis, over 500 cells from more than 200 patients across 13 different cancers were analyzed. In the right panel, you see the contribution of each of the cell types to the various different cancers. Taking this information, Nieto et al. Applied the HindLab's Spotlight pipeline to deconvolute each spot of a Visium transcriptomic assay. In the left panel, on the left side of the left panel, you will see the spots colored by gene expression signature. So we have fibrotic tissue, HER2, or ESR status. In that same panel on the right, you see each spot colored by the overall contribution of the individual cell types. In fact, if you zoom in, you will see that each spot now is represented by a miniaturized pie chart, which shows the contribution of each cell type. This is a wonderful way of visualizing the data and understanding what the component or the contribution is of individual cell types. I hope that you found this presentation to be informative. For anyone wishing to get started with or take their single cell and spatial transcriptomics research to the next level, please take advantage of the numerous resources on our website. Further, if you have any support questions, please visit our support website or reach out to us at support@10xgenomics.com. I'd like to thank you very much from Team 10x and wish you a wonderful day.